Hello and welcome to Principle of Cognitive Neuroscience Part A. During this course, I'm trying to explain to you the main models of how people imagine how the brain works. I will also tackle the main concepts that I usually use to publish papers, what really constitute a result, and we're going through practicals whereby we have hands-on onto how to manipulate data to produce results, how to write a paper, how to get funding, how to code analysis, and how to survive as a student in academia. I'd like also to invite you to the NeuroChino every Monday morning at 9:30 a.m. We organize a NeuroChino with the clinical neuroanatomy seminars, whereby we discuss what is exciting about last week science. Everybody's welcome. You can either watch it on YouTube or connect directly on Zoom to discuss with us. But without any further ado, I'll start with my new lecture entitled Brain Gradients. This lecture is slightly shorter than the others because brain gradients, even though they've been discovered a long time ago, is a new approach of neuroimaging, so it's still relatively less compared to other domains. So I think we can really attribute the brain gradients to the theory and discovery of Friedrich Sanides, who was studying the cytoarchitecture of the brain. That's looking with a microscope at the organization of cells in the brain, particularly the cortex. And as you may know, most of the neocortex is constituted of 6 layers. And these layers are easily recognizable because they have a different density of neuronal cells, and those cells have also a different size. And if you look at it, that really creates those layers. They're not so easy to see. As you can see on the left, this is picture drawing of the different layer of the cotton. You can see that those boundaries are not as sharp as we imagine they should be, or at least not everywhere in the brain. But you can still, by using a computerized method, extract those boundaries. And so by working on the brain, really Friedrich Sanides come out with this hypothesis that there is a progressive differentiation of the organizational pattern of the cytoarchitecture in the brain. And so he went through different portions of the brain and look at how this transition is happening. And right away, he linked it up with the idea that this was a signature, this progressive transition was a signature for brain evolution as we know it. Mainly what he did, he tried to find the origin of this progressive differentiation, and he could back trace the origin of this progressive differentiation to the hippocampus, which has a very simple cytoarchitecture, and to the olfactory center, the olfactory cortex, olfactory center or cortex, which has no layer at all. And those were like the two origin for this strand of differentiation of the cortex. You can see on the right part of the slide from the hippocampus, which is called the hippocampocentric division, you have a progressive differentiation. So you see at the medial section of the brain, hippocampus is coded in H area 28, and you can see how the arrows are showing how the cytoarchitecture progressively changed and created those new regions in the brain that complexified and allowed to build the cognition of the human as we know it. In parallel, and you can see it mostly on the lateral view, the olfactory or olfactory cortex, which is coded here as number 13, progressively radiated and differentiated onto the lateral surface of the brain, creating like new areas that are more complex and building same thing the human cognition as we know it. What's fascinating here is that the olfactory center are probably the earliest way a brain organism can interact with the environment by smelling really what is around it to orient itself toward food and potential reproduction, while the hippocampus-centric center is on the other side the ability for this organism to build from its experience and optimize its survival by learning really from mistakes. And from there, you can add layer after layer after layer of complexification. So this theory, I always found this theory really fascinating. And you can see that through the evolution, those two trends actually meet at the level of the intraparietal sulcus in the parietal lobe and the middle frontal gyrus in the dorsolateral frontal cortex. And those will be eventually, and we discussed that before, part of the neocortex that are the most recently evolved on the evolutionary tree. So during my work as a young researcher, I've been trying to track those trends. I looked at how in the frontal lobe mostly we can see a progressive differentiation of the frontal cortex and found out that actually you have a progressive change in the thickness of the cortex as you're going from posterior area to anterior areas in the brain. And I see that like by using neuroimaging and cortical thickness, as well as a progressive decrease of the myelination of regions, with a anterior region in the frontal cortex that are the least myelinated in the brain, and the posterior regions that are on the frontal cortex that are the most myelinated in the brain. And that will mean that you need fast and instantaneous communication from motor regions to really compute and send the information to the spinal cord and muscles, while actually the more anterior part of the cortex is requiring probably slow implementation and integration of information before sending its process to further regions. I always like to compare that between the a quick young person running and want to always go for any kind of opportunities, which will be the motor cortex, compared to the all-wise human that will take his time to take an answer, his or her time to give an answer. But this is neuroimaging, and to be honest, neuroimaging is not biology, and you still have a large gap to fill in order to really being able to interpret what magnetic resonance imaging is telling us, and being sure really that T1 T2 ratio is myelin. But we can use the information from neuroimaging to try to explore a little bit biology, and this is possible thanks to new tools such as the big brain. The big brain is a resource that has been published in 2013 of a single human brain that had been sliced in very tiny slices, giving you access to the microscopic resolution of the human brain. You can see here I've been magnifying in the Broca area, and we can see those different layers in the cortex, really, which is a grayish area. And you see how you see at least 3 or 4 layers there. And you can actually play with this resource and learn a lot about what is a cytoarchitecture. those slices have been stained with silver staining that we know is able to densely mark about 10% of the neuron of the cortex, which is enough for us really to see the density of what's going on. So if you look at the same progressive transition that happened in the frontal lobe, you'll see that as you're going from posterior regions on the left to anterior region on the right of the frontal lobe, you have a progressive clarification of the staining. So the staining of cells by silver will make some dark staining, and having progressively lighter slices indicates that you have less and less neuronal matter that is stained. So what is going on in there? Less neuronal matter stain will mean maybe you have less neurons and you have maybe more dendrites and axons between these neurons that are taking space and spacing out all these neurons that make like the staining clearer. Something that we also investigate, getting back into neuroimaging by using diffusion weighted imaging. So as you may know, if you've been watching my other lecture, diffusion weighted imaging is a method that is measuring the diffusion of water molecules in the brain. And so what you can see here is that those water molecules are diffusing in an aleatory fashion, but they are partially restricted by the concentration of cells that are inside this tissue. In other words, if the tissue was densely packed with neuronal matter or neuronal cells, you want to have that much freedom from those molecules to diffuse. And the more they diffuse, the more you show that you have interneuronal space whereby you can have synapses and connections, or axons, or just like empty CSF space. If you look at that in details, and you from a posterior to an anterior axis on the stereotaxic space, so still on the right, the most anterior part, on the left, the most posterior part, you can see that this is true. You have a progressive transition, a progressive increase of the ability of water to diffuse as you're going towards the frontal pole of the brain. So in the posterior area of the cortex, water doesn't diffuse that well, it is constrained, and as you're going towards the frontal pole, it is more and more free, which means that you actually have a lot of diffusion and water moving in there, which we might think is related to higher connective property in there. But how can we measure this? So we developed like a new way to look at this, and we thought, well, if you have if you do have more connections, more exchange locally inside a voxel, what can happen in terms of the profile of connection of this region? And so we thought like the overall complexity of the profile of this connection can change. And this can be symbolized as a flute, for example. If you have a region, which will be a flute, that only have 7 or 8 notes, the number of different melody you'll be able to create will be much less than if you have a flute that have way more notes, whereby you can create a lot of more different melodies. And this is important because with a flute on the left, the pattern of melodies that you create might repeat itself more often than the one on the right. And this is quantifiable by using an equation, which is the equation of the entropy that give you a score, which will be a score that reflect how rich your information is in that region, how many different states you can take with that flute or with that region. In order to look at the activity of brain region, we used resting state, functional connectivity, high resolution from the Human Connectome project. And by applying the same concept of measuring the entropy, this is a Shannon entropy, we've been able to quantify how rich really the state you can take in this region, and see that the further you're going towards the frontal pole, the richer the number of states you can take, suggesting that your flute in the frontal pole got more nodes than your flute in your motor cortex. And the number of nodes that you have on your flute are actually the number of connections. All these are examples of gradients that happen in the frontal lobe. And if you take the Oxford language, and I know gradient might not be so easy to understand for all of us, this is that the gradient is an increase or decrease in the magnitude of a property, such as temperature, pressure, concentration, color, observed in passing from one point or a moment to another. Typically, you have a gradient of color at the bottom, where you're going from white to gray and dark, and you have this gradient, changing gradient of darkness that is happening there. So how does it relate to things we can measure in life? So I like usually to take the following metaphor. Think about, I don't know, 1,000 different soup recipes that exist in the world. If you look at it, like you can look at the billions of voxels you can have in a single MRI, you'll see that they all have a different profile. They can have a different color, they can have a different taste, they can have a different smell. You can visually see the ingredient in some other you can't. Um, and this is due to the high dimensionality of the difference you can have. To be honest, it is quite confusing. But what you can do, you can try to order these soups according to main gradients. And so like really the idea is behind the final characteristic of your soup exist several ingredients that are used in different quantities. These ingredients are components and of how much, and you know, how much of these components are in your soup can be arranged along what we call a gradient. For example, the gradient of salt, um, with a less salty soup on the right and on the left, sorry, and the saltier soup on the right. Another example will be the gradient of water content that will be with the thickest soup on the top and the most liquid soup at the bottom. And putatively, if you just took these two ingredients, you can reproduce 50% of the taste of all your soups. And of course, you will need to know all the ingredients to reproduce 100% of the taste of the soup. This number, the 50% and 100%, is what we call the percentage of explained variance. It's how much of your soup you can explain with these two ingredients. As you decompose your gustatory signal in or soup, you know, you can start to find other ingredients that are not in all the soups, like for example, the carrot content. Some soup on the top left do not have carrot at all. And really knowing this ingredient will not contribute at all to knowing the taste. Um, However, the carrot content is crucial to the taste of many other soup on this chart, and adding this carrot content to the list of ingredients allow to explain an additional 10% of our soup taste, bringing it to 60% of explained variance, or 60% of reproduction of our soup taste. Um, but this is an example, and the question really is, how can we extract all these ingredients? It's, do we have to taste all the soup and guess how? Well, you have actually mathematical approaches to do that, whereby you can organize all your different soup onto a correlation matrix if you want, and this is like a scatter plot of this correlation matrix. And the main vectors that will explain the difference between your soup, you realize, is the water content. Still, the thickest soup at the bottom left, the the lightest soup on the bottom left, and the thickest soup on the top right. And as I said just before, you have an orthogonal factor, which is the salt content. And as you can see, with those two factors, you can build a space whereby you can localize each of your soup independently for each other, and probably reproduce 50% of their differences. Um, but life is not always as simple, and you don't always have just a linear representation of all your different tastes. And sometimes some of your vector will not follow a linear rule. And the debate into how to define those ingredients, whether we're talking about soup or brain regions, so we'll come to the brain very soon, um, is whether a linear model will be enough to extract all the information that we need, or do we need to push our model to nonlinear models? The great advantage of linear models is like they are simple, and you can do a lot of manipulation with it that you will not be able to do with nonlinear because they are extremely complex. Um, Here you have an example on the left of a two-linear approach. We have non-negative matrix factorization and principal component analysis. So that's the top and the bottom on the left, applied to thousands of different faces. You can see that non-negative matrix factorization will identify different components of what constitute a face, and will be able to, thanks to this component, to recreate a face based on the decoding of this face based on these ingredients. Similarly, you can see that the principal component analysis not only extract ingredient, but also extract negative ingredient, because you're going two sides, so you have positive and negative contribution. And by using all these components, is also able to reproduce and decompose a given face along these dimensions. But you also have many nonlinear approaches that are able to do the same thing, and which putatively should explain more variance than the ones that are linear. They have a kind of barbaric name. You have Laplacian eigenmaps, diffusion map, t-distributed stochastic neighbor embedding, and uniform manifold approximation and projection. Those are only 4 examples of the many different approaches that you can have. And along my lectures, I have been using some of these methods and didn't mention, and we're just going to come back to some of the results I presented to you, and I'll bring it back into those different nonlinear approaches, so you can really see. Principal component analysis, for example, was the division that I showed to you before of the frontal lobe. It was based on the principal component analysis, didn't need anything more than a linear approach to do it. But another approach was the approach that been really pioneered by Leonardo Celiani, who used Laplacian eigenmaps, which is a nonlinear dimension embedding, to find components of connectivity that he projected back onto the insula of the brain. And he found that you had a gradient of change into the profile of connectivity that you can explore on the insula. That's like quite a fascinating approach. And so you can see here the matrix on the left is a correlation matrix between the profile of connectivity of the different voxel in your insula, and then you extract the main component using Laplacian eigenmap, which I told you just before is like a principal component analysis, but nonlinear. It's a nonlinear dimension embedding approach. And so you can plot them into this 2D. As for the soup I showed to you before, you can see that they are very well flattened and organized along a first component with a little bit of differences that can be found on the second component. And then you can project back these colors that you have onto your one component and two component scatter plot onto your insula, as you can see here on the right panel of this slide. And there was really a pioneering approach of the use of nonlinear dimension embedding and projecting it back onto the brain. Later on, we use exactly the same approach to study the lateralization of the brain. You remember we had all this map of functional lateralization according to different terms that we reorganized together using Laplacian eigenmap to get the main axis of differences, the main ingredients that make the lateralization of brain functions that you can see here. And those functions were organized according to 4 axes: an axis of symbolic communication, another one dedicated to emotion, another one dedicated to perception and action, and finally a last one that was centered on decision making. Now, I also spoke about T-distributed stochastic neighbor embedding, and there we coming to my lecture on epistemology, whereby we took 1,333 strokes and their disconnection. And the stroke will be on the left in C, the disconnection will be on the right in D, and we plotted them into the same space. And we added as well fake lesion to see whether the distribution of real stroke was biased in terms of the spatial distribution of stroke was biased, also disconnection in stroke was biased. And you can see here that you can summarize the differences that you have between your different stroke, whether you look at the lesion or you look at the disconnection, into a 2-dimensional space to make a map that is informative and in which you can actually guide yourself to find differences and similarity across your cases. That brings me to the diffusion map, which is also another approach that's been used and applied to functional connectivity by my dear friend Daniel Margulies, who used as a diffusion map to extract main components of functional connectivities, and mainly find like 5 dimensions that he called gradients, or those are the ingredients, if you want, of a functional connectivity. And the first and the second one really capture his interest. And really, this component have been of main interest for Daniel Margulies because it seems to kind of bring together the models that Marcel Mesulam have been describing, whereby primary areas like a cluster separately and gonna bring together to like a multimodal conceptual regions that are in the heteromodal paralambic and lambic regions that you can see here. You have V1 and A1, primary auditory and primary visual area, and they constrain themselves together towards the limbic and paralimbic system. And what he has when he's doing this mathematical representation of functional connectivity, he got this first gradient and this second gradient, and you can see that they kind of merge together toward the red regions that happen to be in the heteromiddle and paralimbic and limbic regions. And you're really going from unimodal regions to the most abstract multimodal high dimensional region where you're going to combine together a multi-sensory input. He did that in human, and he reproduced his finding in monkeys by using directly axonal tracing, and found that you have the same gradient whether you use functional connectivity in humans or axonal tracing in monkeys, by doing so promoting really a comparative and evolutionary perspective to his gradient. But further than that, if you have those regions and you start decoding their contribution along the gradient, you start really showing how different percentile on the gradient, which like how strong are you on this place on the gradient. They're just going to come back and show you that's like how far you are on gradient 1. On, you know, if you 6, you're very high on gradient 1. If you -4, you're very small. And this is converted here in percentile from 0 to 100. And you can see how those percentile load onto different functions that are really going from unimodal idiotopic areas to the most advanced multimodal integrative region. So you're going from motor, eye movement, visual perception to autobiographic memory, verbal semantic, emotion, and social cognition that are not, you know, that are the integration really of the input from a motor eye movement that you got, and like you're gonna integrate and process it, and like the highest level of function at this level. Um, and then since you have those maps, you can use it and use the same percentile approach to create connectivity matrices and project back this level of functional processing somehow onto the corpus callosum. So you have the gradient in A, you have the different representation of the different percentiles that we just spoke before on the slide here. So you have, you know, like those 3 different level of percentile, which are an example. They have different connectivity, structural connectivity profiles that intersect differently in D with the midsection of the corpus callosum, and that allow you to project back using a weighted average the values of the cortical gradient onto the midsection of the corpus callosum. And you can see that you have a U-shaped relationship whereby they're really the highest, well, most unimodal interaction in the brain are happening in the posterior part of the body of the corpus callosum, and the anterior part of the corpus callosum actually contains the highest level of computation in terms of model and high dimensional processing. You can also like try to segment the corpus callosum according to this different level, which has an added value for neurosurgery. So in other words, if you take if you sample different point onto the corpus callosum, you would see that they have different profile of contribution onto this percentile representation. And this you can put it into a matrix and apply an algorithm which is a little bit like k-mean clustering, but a bit more advanced, which is SGB scan. And then you can divide your corpus callosum according to the different profile of level of relationship with the principal gradient of the corpus callosum. This principal gradient really link up the limbic system, which emerged from the hippocampus that we said with the rest of the cortex, mostly on the medial portion and then projecting on the lateral portion. In other words, the first gradient of the functional connectivity really look like the hippocampus-centric system. But there is no word about any gradient that will be related to the olfactocentric system, which is the second trend of the dual origin. So in the following studies that I'm going to show you, we try to assess whether it was true that the first gradient will be specific to the hippocampocentric system, keeping in the idea that the hippocampocentric system has to be related to the memory of episodes or episodic memory. So we'll run a correlation between this and episodic memory activations. And we'll try to explore the other gradient of the brain to see whether one of them will be related to the olfactocentric system by just trying to relate it to functional activation that are associated with olfactory input. And this is what we did in this paper, and you can see that in fact this is true. The gradient number 1 is really related to functional activation that are consequent to the achievement of episodic memory tasks. It is significant, and this is the only one that is significant. And in contrast with that, the gradient number 5 is the one that is related to olfactory tasks. So you really see how you get here two gradients that seem to match a theory of the dual origin. Then the question is to explore the functional role of gradient 2, 3, and 4. Gradient 2 is really visual, but its origin is kind of unknown, and 3 and 4 remains to be investigated. Reading the brain in terms of gradients also gives you the unique opportunity to bridge the gap between species, and this has been beautifully illustrated by this study by Ting Zhu. What they did essentially, and they put together in the same matrix human and monkey functional connectivity. So in other words, on the top left red square, you have the connectivity of human with themselves. They have a specific profile of connectivity. And in the yellow square, you have the connectivity of monkeys with themselves. But at the bottom left and top right window, you have the connectivity pattern of connectivity between humans and monkeys. And there you can actually investigate a similarity in the profile of connectivity by using a joint embedding. I think it was a principal component analysis here. And so you see how you compress your dimension in a few components. And these components, just gonna have my activation here, can be this component can be projected back onto scatter plot. Here you have a two-dimension scatter plot that correspond to the component number 1 and the component number 2, and that you can do in the human brain on the top and in the monkey brain at the bottom. And then you can repeat this and project it onto the surface of the human brain and the surface of the monkey brain, whereby the color codes you see here show the areas that have an equivalence along these two dimensions in, along the first dimension in human and macaque. Then you can repeat the same experience with the second dimension. And here you see along the second dimension, the areas that have an equivalence between humans and macaque. And then you can try to have a look at a third dimension and again reproduce what I just said to do by projecting it back onto the brain of the human and the macaque. And you can see by this color coding the areas that share similarities between species. And like if you put together all these dimensions, then you can parcellate really the surface of the brain into several regions that have to be comparable across species. And by doing so, yeah, this is really bridging the gap between the two species by using a functional connectivity. This is absolutely great. And then you have a match. That's a beautiful illustration here. You have a match between the human division and the monkey division here. Next, really, you can also use those gradients to investigate more about the origins of the brain, and by doing so, you can investigate like the contribution of subcortical areas to those gradients. And this is what has been done here. And instead of looking at the local representation of this gradient, you try to make a link between different voxels of the brain by creating a tensor, like a little bit like tractography, uh, uh, between voxels that share similar but slightly different profile of gradient. And by doing so, you can actually visualize like the progressive transition in the magnitude of the gradient in the brain. This magnitude of gradient across the brain sometimes have a slow transition and sometimes has a very sharp and quick transition. And this approach allows you to actually segregate in the brain the different structure by just looking at the profile of gradient magnitude. And then you obtain a division of the brain, of the subcortical region into different areas just based on the profile of functional connectivity, on the profile of transition of magnitude of gradient. And because this transition of magnitude of gradient is not the same between each region, you can start ordering it into a different level of segregation. And this different level of segregation from scale 1 at the bottom right to scale 4 are different level of differentiation along evolution, or suggested being different level of differentiation along evolution. So to repeat that, you see on the top, you have this hierarchical division, progressive diffusion. Every level is a level of complexification of the division of subcortical cortex. And at the bottom, you have like a representation of this division at 4 different levels: scale 1, scale 2, scale 3, and scale 4. And those 4 different levels of differentiation putatively will be different level of differentiation along evolution. Now, I know what you're saying to me is like, okay, this is great, but what can I do that could be really fun with that? Well, what you can do that will be really fun with that will be to start looking whether the principal gradient are the same between different population of people. Let's take those two different population, a population of LC control participant and a population of LC control participant, except from the fact that they just took LSD before getting inside the scanner. As you can see, you can decompose a gradient comparably, and they kind of look the same if you just look at it real fast. You have a first and a second gradient that are localized in the same areas, whether you took a placebo or if you took LSD before going in the scanner. If you do the scatter plot embedding of the first and the second gradient, you can see also that you have a distribution of these networks that are kind of similar between people that are under placebo and people that are under LSD. Although the distribution of the different point in the brain seems to be a little more constrained in the people who took LSD when you compare it with people who took placebo, indicating that LSD might have changed the way your brain region discuss during the scanner. If you do a simple comparison between LSD and placebo, you will see that actually, as you take LSD, you will increase your magnitude of the principal gradient, the gradient number one, in your medial limbic system, which is all about, introspection, memory, representation, mind wandering, and you will decrease your second gradient, which is the link between your visual areas and the rest of your brain. This will suggest that as you're taking LSD, you get way more into your imagination and travel into your thoughts and way less taking information from what you actually see, and actually what you see is only coming from inside your brain. So this is done for the lesson on gradient, and I'm waiting for you to listen to my next teaching, which will be focused on precision medicine. We'll speak a lot about statistics and the way to do this. This is also like a kind of future approach. Um, I would just want to remind you that the evaluation of this course is based on participation, attendance, and a little project, which is writing an essay. The deadline is December the 11th. It will be dedicated to the subject of brain hierarchy. I want you to choose a hierarchy. Give me a general introduction, explain to me the anatomy of this hierarchy, tell me whether this hierarchy is lateralized or not, and if you don't know, don't find any reference on how this hierarchy would be lateralized, how would you measure the lateralization of this hierarchy? I want a section on brain evolution, link this hierarchy to brain evolution, and a synthesis part whereby you've wrapped it all together. All that in 1500 to 2500 words, not including the reference at the end of your manuscript. To be sent before the December 11th midnight to my personal address, michel.thiebaud@gmail.com. I thank you very much, and I'm looking forward to seeing you in the next lesson.